Many frogs lay terrestrial eggs relying on environmental conditions for embryonic development. These eggs are vulnerable to dehydration, which may alter development or cause mortality. Drying-induced hatching is known from several lineages of frogs that independently involve terrestrial eggs, deposited in gelatinous egg masses on leaves above water. However, we don't know whether these or any terrestrial frog embryos hatch early as a direct response to high temperatures, showing a behavioral thermal tolerance limit or VTMax. The expression of a VTMax enables animals to avoid exposure to temperatures closer to their physiological thermal tolerance, that is, their critical thermal maximum. It has been studied in lizards and recently in anurans and can be affected by diet cycles and dehydrations. Red-eared tree frog embryos hatch early in response to multiple threats. This species' robust Q-hatching response offers a good model for experimental studies of embryo behavior. We use a heating experiment on this species to assess if the hydration level of clutches and eggs affects the response of embryos to warming. We suppose that embryos can hatch early in response to warming. Dehydration reduces the VTmax of embryos, and fully hydrated jelly protects embryos from high temperatures and dehydrations by slowing the heating of clutches and eggs. During 2 rainy seasons at the Smithsonian Tropical Research Institute in Gamboa, Panama, we collected wrestling needle clutches from experimental ponds. We topped them to an ambient temperature laboratory and randomly assigned clutches to wet and dry treatments until age 5 days. That is, midway throughout their plastic hatching period when they are highly responsive to environmental cues and also highly discriminating with few embryos hatching spontaneously. We tested embryos' response to heating in 2 different contexts, in intact clutches and in small groups of eggs insulated from jelly and leaf. For heating clutches, we use 6 hydrated and 8 dehydrated clutches. We set up them for experiments at age 4 days on an individual support stand with a scale for subsequent measurements, placing 2 temperature recorders inside the jelly, red dots among the eggs, and 2 more touching the surface of the leaf. At age 5 days, we recorded egg and clutch measurements and observed embryos for 1 hour at ambient temperature to measure a baseline hatching rate. Then, we heated each clutch, controlling the heating gradually and continuously without exposing the embryos to heat shock, thus preventing them from reaching their CT-max quickly. For each embryo hatched, we recorded the time and the temperature of the nearest thermocouple. We stopped each heating trial when the clutch reached 40 degrees because the highest rainy season temperature recorded so far at the experimental pond is 39 degrees, and exposure to 41 degrees was lethal to embryos in our pilot observations. Finally, we took a photo for final measurements. For heating eggs, we use eggs from 5 hydrated and 7 dehydrated clutches. We removed eggs from their clutches just before the onset of mechanism-sensory Q-hatching at age 3 days. We distributed 4 thermocouples red dots among the eggs to record temperature and placed a scale. At age 4 days, we controlled hydration manually to maintain the desired egg size and hydration level. At age 5 days, we recorded the initial measurements. We positioned a camera in front of the assembly with a time-lapse controller to photograph the eggs every 2 minutes. Then, we recorded the baseline hatching rate as explained above, and after that, heated assemblies. When an embryo hatched, we recorded the time and the temperature of the nearest thermocouple. Trials ended when all embryos hatched or the temperature reached 40 degrees as above. All individuals were monitored for 24 hours to determine survival after heating. Then were released to the tadpole at the experimental pond. We found difference between wet and dry eggs and clutches. At the start of trials, eggs were smaller in dry clutches and dry eggs than in wet clutches and wet eggs. The mean duration of assays for dehydrated clutches and eggs was less than for hydrated clutches and eggs. Only 2 clutches, both dehydrated, had complete hatching before reaching 40 degrees. In all trials, all eggs had hatched before reaching 40 degrees. The baseline hatching rate at ambient temperature just before heating trials was much lower than the hatching rate during heating for both hydrated and dehydrated clutches and eggs. Hatching rate was better explained by heating hydration structure on all interactions. However, the only significant effect was the heating by hydration interaction. Heating increased the hatching rate and did so more strongly under dehydration in both structural contexts. Wet clutches had a higher mortality than dry clutches or wet and dry eggs. The best explanatory model included hydration, structure, and their interactions. However, only structure has an effect on the mortality of the embryos during heating. Both in intact clutches and eggs, dry embryos hatched at lower temperatures than well-hydrated ones. The VTMax was better explained by hydration, structure, and their interaction, all of which were significant. In clutches, wet embryos expressed higher VTMax than dry embryos, and wet embryos in eggs had a higher VTMax than dry ones. However, the effects of hydration on VTMax were weaker in clutches, with substantial overlap across hydration levels. They were stronger for eggs without jelly. All embryos who expressed their VTMax successfully survived 24 hours after heating. During development, warmer temperatures can produce smaller individuals, increase development rates, heat rates, and oxygen consumption, and decrease hatching success. Here, red-eyed tree-frog embryos express a VTmax and escape from heating by early hatching. The expression of a VTmax occurred on a specific temperature range, depending on hydration state. This reflects an immediate decision to escape not only from heat, but from other interacting factors such as hydration state. These results support our hypothesis and demonstrate accompanying behavioral assay to elucidate the responses of frog embryos to stressful conditions of temperature and dehydration. Egg loss volume during heating. How much was best explained by hydration, structure, and the interaction, but only hydration had a significant effect. During heating, hydrated eggs, both in clutches and insulated from jelly, lost more volume than dry eggs, which started out smaller. Water loss from wet eggs and hydrated jelly slowed temperature rise, and the hydrated clutches and dry eggs heated faster than hydrated ones. The mean heating rates during heating trials was best explained by a model including mean egg volume loss, hydration, and their interaction, all of which were significant. There were no significant effects of structure. So, hydrated eggs and clutches have more thermal buffering capacity via evaporative cooling. This supports our hypothesis and indicates that the presence of jelly confers hydric buffering during heating, delaying hatching in terrestrial anurans. Hydrated clutches will still have a core of jelly, and the eggs have moved to the surface of that jelly, so they still could absorb water from the jelly. However, with more frequency of dry days, the jelly will have little water to be absorbed by the eggs, so the size of the eggs decreases, constricting the perivitelline space. Thus, the amount of water stored inside the eggs is particularly important for thermal buffering. Heating-induced hatching may serve as a non-lethal method to access for embryos' vulnerability to changing climatic conditions. Warming and dehydration clearly interact to affect embryo behavior. Terrestrial egg clutches can be affected by lower frequency of rainfall. Rainfall prevents dehydration, and hydrated clutches can buffer and withstand higher environmental temperatures. Future research could help to understand if the VTmax of the embryos is affected by other experimental and biological factors. This study could also enable large comparative studies to elucidate the response of anurans in their early life stages to stressful conditions of temperature and dehydration. Thank you everyone for assistance, and thanks to the institutions that funded and still funding this research. Thanks to my advisors, and thanks to Team Tree Frog for file assistance, comments, and suggestions on this work.